All right, good afternoon, everyone. Thank you for coming out this afternoon, and thank you to the conference organizers for having me speak today. As I was introduced, my name is Kashef Qaadri with a company called Biomatters, which is a software company. Just a bit about myself. I have a background in biology, more specifically microbiology, as well as neurology and molecular genetics. And over the past 15 years or so, have been in this field of bioinformatics. So the title of my talk is an intuitive 16S rRNA biodiversity tool, which I'll spend the next 12 minutes or so chatting about. Just in terms of an overview of the talk, I'm going to provide a really quick introduction to the company I work for, Biomatters, as well as just a bit about the software, or more specifically the lack of software that exists for 16S biodiversity, followed by a use case looking at the Sorcerer to Explore project, which is a really interesting project which we'll talk about, and then just wrapping things up with a workflow, which is how to scale these types of biodiversity projects from just a single sample or a single set of samples to something much larger and more scalable. So in terms of the introduction, I'm not sure if any of you recognize this map off to the right, but it's the London Tube map. And if you're familiar with London or any mass transit center, if you have a question like, where do I find lunch, you need to understand how to navigate from point A to point B, but in an efficient manner. And the London Tube's a really good example of having things very prompt and very scheduled so that you can get from that point A to point B. Now, what's the connection between the London Tube and biology is some of you may have seen this, which is a slightly off, but 16S map, right? And it's much the same way, right? So when biologists want to understand the biodiversity of a sample, they bring a lot of contextual information to that, something that can't always be translated to biologists or bioinformaticians. So that's one big problem is just bringing in contextual information, the background information, what are the specifics, what are the nuances to a specific project. The second is that bioinformaticians are dealing with a lot of volume, right? They are typically very understaffed. I'm not sure how it is in your country or your location, but in the States, there are very few bioinformaticians and a lot more biologists. And so it becomes difficult for the bioinformaticians to deal with the masses of data, and to add to that is this issue of the contextual information, right? So we've taken an approach of letting the biologist do a lot of the interpretation and analysis themselves, or at least the first pass of that. And that way the bioinformaticians can focus on areas that are more difficult, the harder problems, the things that actually require algorithmic development and a little more of their proper skill set. And of course, because of the overload of bioinformaticians, or overload of data that the bioinformaticians have to deal with, the time that it takes to get your results back is often long, and again, this speaks to the biologists being able to do it themselves, because then they don't have to wait, they can just do it themselves. And thirdly is the issue of visualization. So biologists, so I'm a biologist myself. I have been doing bioinformatics for a while, but consider myself a biologist. And I always like to see things visually. It helps me understand what's going on, and I always like having a GUI, graphical user interface, as opposed to anything command line driven. And so Biomatters Biomatters is a New Zealand-based company established back in 2003. We have an office in San Francisco, which I work out of. We've been around for over a decade producing the Genius software, and across the globe, 100 countries and several thousand customers in that space. And it's a bioinformatics platform that runs on a Mac, PC, as well as Linux machines. And the main focus of Biomatters is to provide tools for NGS analysis, molecular biology, and phylogenetics. And over the years, we have been cited and used quite a bit. We are now the most cited commercially available software, and we really pride ourselves on the number of publications that are being accepted using our software for the analysis part of that. So in terms of software that's available, this has become a really big problem because really there is none for, no real good tools for 16S analysis. Most of the tools that are available come in two flavors. The first is things that are command line driven, which is a huge problem for most biologists, right? So just trying to script and hit things programmatically is a big issue. And there are programs to do it, but again, the barrier to entry for most biologists is quite high because it is command line driven. And the alternative is to use a web-based program. There are a few of these available. There are very strict limitations in terms of what size file you can upload, how many files you can upload, the speed at which it processes, and sometimes there are even privacy concerns. There are a few products on the web that are available, but they require you to give away permissions to use the data in other cases, which isn't always a good idea. Excuse me. So today I wanted to use an example which is called the Sorcerer 2 Expedition, and it's a biodiversity workflow, and this is going to be used to highlight the tool. So I'm not sure if any of you are familiar with the Craig Venter Institute, but they commissioned an expedition around the globe to collect water samples, 200 to 400 liters every 200 miles, and they did this expedition essentially crossing the entire globe. And the idea was to take these water samples and filter them with progressively smaller filters and collect microorganisms to see the biodiversity of microorganisms in water. And as we know, the importance of microbiology is that you need to understand the ecosystems, the biodiversity, both on land as well as sea, to get a really good feel for how our ecosystem is put together. So again, they were collecting aquatic bacterial communities using this filtering method. They would be freezed and then shipped off for sequencing to either the JCVI Institute in Bethesda, Maryland, or in San Diego, California. And so just in terms of the workflow, is collecting these samples on this expedition, sequencing this back at one of those locations, and then analyzing the data as they became available. And then this is just a map of the expedition, and again, you know, the main thrust of this was to collect the samples and then 16S rRNA, and then to figure out the species structure as well as diversity of the environmental samples using fragments to figure out metagenomics. And not sure if you can see in the back, the blue line was the original expedition, which went from 2003 till 2008, and the black line there is the second route, which was 2009 to 2010. Did a really good job of capturing a lot of the biodiversity as part of this. So in terms of the analysis, the first part is pre-processing, which is binning based on the MIDs, or barcodes, which is shown in the diagram off to the right. So this can be done within the Genius platform, which is, so the image you're seeing on the right side is a screenshot of that. The second is the trimming part, and so this can be done two different ways, or several different ways, two of which are highlighted here. One is to trim the sequence based on quality. In this case, I've selected 0.05%. And the second is to trim out adapters or based on primers because they are multiplexing. And the third is to remove chimeric sequences. This can be done a number of different ways. In this example, we used a software called Chimera Slayer, which identifies the highly likely chimeric sequences and then filters them out. So after the pre-processing takes place, we use this tool called RDP, the Ribosomal Database Project, which has the ability to analyze your data online. And this can be used with both the full 16S sequence or fragments of that. Obviously, the quality is going to be much better if you have the full sequence as opposed to the fragment. And there is a limit of 500,000 sequences, so 500,000 sequences that are available, or up to 500,000 sequences at one time. And again, this is looking at both bacterial and archaeal bacteria, or sequences rather. So in this case, this slide is highlighting the sequences which have been pre-processed. So once you drag your data into Genius, you can do the pre-processing, as I mentioned earlier, with the barcoding, trimming out primers and adapters, as well as trimming out low quality reads, and then taking out the chimeric sequences. So the data would look like this. And then you can actually begin the analysis process. So the first step of this is to submit your job to RDP. This again is completely transparent, in which you just enter a name, which is highlighted here in the screen to the right, which has a job description, so that's the title of your project, and then just simply enter in your email address. This then is uploaded to a very secure web server in which your data are processed, and then I'll show you in the next slide what the end results are. Just in case you are curious, this is included as part of the Genius package. There are a few different hyperlinks there which are in blue. So these include the terms and services as well as the privacy, and you can click on that and read how we ensure security and privacy of your data, as well as the requirements to use the service. There is also a demo and help manual there. So the results of this are highlighted here. This again is a secure web link. And the results look like this. It's an interactive HTML page that's returned. And that gives you both the species classification as well as the relative abundance of a particular genus. And this can be scaled up using a workflow builder, and you can certainly include the 16S portion of that within your workflow. So again, what I talked about was the RDP, the classifier project, using the full length or 16S from the pre-processing, the analysis, and then reviewing the results. And with that, I'd like to thank you all for your time and open it up to questions. I do have a booth in the exhibit hall just as you enter to the right, and we are giving away free t-shirts. So I'd love to chat with you about your 16S project and bioinformatics in general. And with that, obrigado, and open it up to questions.
Thanks a lot, Kashef. I think we have time for one quick question, if anyone has one. No questions? So I think, yeah, as you said, she will be around just in the next building, so you can ask anytime.
That's right.
Yeah.
Thank you.
Approach of quantifying the phylogenetic